right? All right, well, welcome everyone. Thank you for making the time. This is probably the most out-of-the-box session. Yeah, I'm sorry. This is probably the most out-of-the-box session. When Agnes and I, Agnes is one of the co-speakers here, we were choosing a theme. We didn't know that things were going to go sour this badly at this time. So now this session is really important for all core facilities. Please be creative. Please make sure that your core counts in the future. Now, what I should tell you is I'm not going to teach you. We're not going to teach you creativity. We're just going to remind you that all of you have it in you, okay? And we're trying to make it as light as possible, so at least everybody can remember something, okay? And so the session today, we have 3 speakers. I have 2 great colleagues and friends here. Agnes is going to talk about 2 impacts, the impacts of one and on the other. And Francis is going to speak about antibodies, and an antibody core can be really creative, okay? And later on, we'll have some discussion here, and we can try to answer any of your questions. So I'm going to talk about, so first of all, hold on a second. Alrighty. First of all, I'm going to talk about, creativity in general. What's creativity and how do we go about it? And then, there are, I need somehow. Need a pointer? Maybe a pointer, maybe. All right.
Let's be creative.
We're going to be creative? Don't kill yourself, please. Right. It's not worth it. So, where is the pointer? Is it? Okay. So, so there are ways to provide the service and to be creative in doing so. So this is the simplest definition. There are hundreds of cores, all of you, and most cores stick to what they know and do best. That is great, of course. In this day and age, and looking around you, doing better, we have to do better than great, and that requires creativity. You have to be creative in many ways to bring something that is above and beyond. So I'm going to attempt to remind you that all of you can be creative. And if you cook and you watch cooking channels, Martin Yan, his motto is, Yan can cook and so can you. So this is, to me, I apply it to everything I do. So before I go too far on creativity, I just asked Google, what's creativity? Okay, and Google says, so the use of imagination or original ideas, especially in the production of an artistic work. So it's almost creativity is in art. In the Britannica, it says, creativity, the ability to make or otherwise bring into existence something new, whether a new solution to a problem, a new method or device, or a new artistic work. So there is some science in there, but it's not totally. Now, creativity is a disposition involving both the ability and the motivation to produce things. So there is a certain effort in there. And then, for me, in my life, I've seen 2 instances where a manuscript comes back from a reviewer, and the reviewer says, a truly piece of art. So now science is an art. Yeah, so that's the highest compliments you can get when you're trying to publish. So everything is sort of connected. Now, there are many among us who will try to study creativity. So, there are apparently more than 100 books on the subject. And there's creativity in research and development. There's interdisciplinary, multidisciplinary. There are theories and themes. There's even a handbook. And these can cost you, anywhere from around $56 to something like $233. And so, but I try to, go further. No, this is not. So when I go further, there is I say there's creativity in everything, and there's creativity techniques, there's creativity on demand, the science of creativity even, and there is even a cure from creativity. You understand? So all of these, you can find them. Perhaps the funniest I find is on my favorite comedian, John Cleese, the Monty Pythons, and he has a short and cheerful guide, and which basically says, many people have written about creativity, but although they were very, very clever, they weren't actually creative. So there's a fine line, and I like to think that I'm writing about it from the inside. That's totally John Cleese, all right? And so what I'm trying to, tell you, just a few facts of life, nobody's born creative, and I say that necessity is the mother of all inventions, and this what basically pushes you to create and pushes you, because the status quo is just not enough. And creativity also is not a private business. It's not actually only few people are creative. It's everyone can be creative. And then if you look in your household, your house chores, you can be creative in doing them. You can be doing your family chores, and then in every work of life, essentially. Now, science is not different, and core facility work is no different either. So essentially, it's just to bring you to what you do best. Now, very simple facts. So the making of a core, so is a group of highly specialized people who provide a specialized service with predetermined boundaries for predetermined set of fees. So that's what we do. If you just, you can live like that if you want. Core facilities cannot write independent grants. This is, as a PI, you cannot write a grant. This is, at least at Memorial Sloan Kettering, if funded, you have to work on it. And then if you work on it, you take resources from others, essentially. And then we had our former president and former NCI, former NIH, Harold Varmus. He says there are 200 plus institutions that allow you to do your independent work. So that's not for cores, essentially. However, as we're going to see later, you can write R01s with other people. You understand? But co-PI, or I call it enrich the people you inherit from. The more people have money, the more you have work. So this is so a typical principal investigator works on a single theme with branching. A typical core facility works on dozens of themes in a single year. So essentially, our advantage is we have access to every colleague's interest. We have access to the deep science, and then we can actually act on that. Now, the beginning of creativity, so for me, at least the way I do it, is essentially you ask yourself the question, how can I make it better? And how can I help them here, all our colleagues in an institution and outside? How can I help them do what they do better? And better could be faster, cheaper, and most importantly, more accurate, high quality, on time, on target. This is what people want. So they want, if you can, do this novel stuff without breaking the bank, without them letting go of a scientist or a technician, you succeeded, provided that you provided the right solution. Now, who will pay for creativity in a core? We cannot use resources for our own. So you need the creative colleagues, the creative collaborators, and shared grants. So it used to be people, they apply for grants and they say, oh, I'm not going to use chemistry anymore. So luckily now, NIH, if you have money assigned to you, you get it. So it is possible. Now, the reality is novel work is not always successful. You understand? So you have to be careful. This is pretty much like experimenting in the kitchen. What you thought, you may not eat what you thought was creative. You may go just to the garbage can. And a creative dish may still need, more tweaking. It's still not edible. So it needs more work. So creativity requires patience and additional testing. So it's an ongoing, reiterations after reiteration. Now, you need more money for development. If you want to develop something, somebody has to support the idea and then support the risk. Nothing is 100%. And so one should have a reasonable plan and a budget. So before you engage in creative things with a colleague, you have to say, well, this may not succeed. This will probably has a great, chance to fail. And are you with me in that? Now, there are two things. So people want A, and then you're proposing B. And as I said here, A, B may not work. So the way we do it in my lab is essentially you have to do both at the same time. And you have to execute what is known. So somebody actually have to take it from your hands and work on it, or take the results. And you have to execute what's agreed to creatively, and then it has to be done in parallel. And I always say that two related projects run in parallel are only slightly more expensive than one. So if you imagine you have twins versus one, so actually at least you share one babysitter. You understand? So it's already you have, so it's a lot of effort. It requires a lot of organization and focus. And, but it's feasible, we do it all the time. And so if creative work shows promise, it will provide grounds for, what you want is to show a proof of principle that it starts working, okay? And so you can have funding, internal or external or both. You have a new direction in research. You have most likely high-level publication. If it's a true discovery, then you have an invention attached to that. And when you say a patent application, most of the time, I have to remind my colleagues, please don't disclose that structure. That's a patent. What do you mean patent? Well, this is novel because I do the background searches, I do what I have to do, so that actually they realize. And then sometimes you sacrifice what I call a dumb molecule for publication, and then the real stuff stays, stays confidential until a patent application is filed. If you even more lucky, then you get the stuff is licensed, taken off your hands. You're founding a new biotech, a big company buys it. So, and then that brings royalties for both institution and core inventors. And I think institutions are trying nowadays to even possibly, I'm hoping, that they bring some money back to the core, part of the royalties. So now, the recipe for me that works is essentially be curious about your colleagues' work. Attend faculty research in progress meetings. It's not published yet. It can be done better. It can be improved. So all of these essentially. Then I ask the question, what could be done better? Hey, I can do this for you if you agree to it. I do the research, the background, the feasibility, I do them. And I share the findings with my colleagues. I send emails. I create a box. If it's too much information, I create abstracts for somebody who doesn't have time. Okay, this is what you're supposed to do. And essentially then, if they agree, sometimes we have 2 or 3 meetings before they agree to something. And you may have to write a grant. Before coming here, I submitted an internal grant. I'm writing a second one now. Most likely, I will get a share of it, but if I don't do it, I don't get the money. You understand? So essentially, you have to do that. And then grants nowadays, you have to have 3, 4 grants, or even some people say 10, and you may have 1 or 2. So So it's a continuous process. You may have to write the manuscript, and you may have to write the patent. So this is all on you to push things through. And now, from my own lab, so basically it was a simple request, scale up a chemical entity, make a kilogram of it, and very quickly, I remember clearly, so this was done 2008, 2009. There was some, it's chemistry, so there are some unstable steps. So when I asked them, I said, did you experience, a little pop or something? He goes, yeah, we had a pop. I said, if I make a kilo in the Upper East Side, it will be a boom. You understand? So basically, you had to make it safer, fix the chemistry, make it cheaper. So all of that, happened, and which led basically to a patent application. During the scale-up, a most bioavailable crystalline form, so this is a polymorph, was discovered, and then which led to a second patent. And the drug basically is now called Erleada, apalutamide, and it's basically FDA approved in 2018 for prostate cancer. And sales are currently about 2.5 billion, which means Memorial gets a chunk of that yearly, and then it probably is going to go, I don't know, 2030s probably. So it's basically, had I not, fought for it and went out of myself, to get things done, it would have been lost. So basically, this could have gone the other way, of course. Dropped the project because people said, oh, this is, too complicated. It requires a lot of money, more work, more development. So essentially, the PI could have stopped it right there. But it took, several meetings, and then I proposed several things how to resuscitate the project because it was actually dead. A convinced technology development, these people said, we will not patent it, So then fights until I got the second opinion, and I made a case. So the whole thing, nothing is given. So we provide the GMP that went to phase 1 trial. We made, so my lab makes GMP grade syntheses. And so the product was licensed, and then the rest. So I think, I'm convinced, and Agnes and Francis will tell you the same. Core facilities should be creative. Core facilities are the most fertile ground for creativity and for science. Colleague requesters should help you, find the ways to be creative in developmental work within core facilities. A creative core benefits the whole institution, benefits other institutions. And core heads should continuously look for ways to fund their creative work in collaboration with principal investigators that use their core. I mean, an idea is an idea until it's done. If you don't do the idea, I could have thousands of ideas like that. But you have to carry it, and you have to make it happen. And so it's never easy. It takes hard work. And sometimes I do borderline harassment. You understand? So it's like you have to stay in people's face. How can you not do it? You understand? This is like, so, but it's for the science, okay? So this is my acknowledgement slides. There is no creativity without an institutional support. That's gigantic. MSK is beyond. I mean, a totally renovated floor, brand new equipment, all that. So NIH and NCI through the core grant, and then I have an R50. We have the experimental therapeutics program at MSK, which is also funds translational projects. We have a lot of, I have the privilege to have a lot of smart colleagues who come with really smart, gigantic ideas. And then, so to me, there's like, somebody says, I have this problem. And I remember one of them is like, he dropped the pen. Like people drop the mic, in all this forever. He dropped the pen and walked away. That's, to me, it's, fix it, essentially. He said, and then, And so I work closely with about 8 core facilities in a cycle, essentially, or in a very give and take sort of way. It goes from clinical affairs to, antibodies to a lot of, so when you're on to develop something for the clinic, there is a whole well-greased wheel that takes over. And people put Gantt charts and they tell you, this is gonna go to clinic in 1 year, 12 months, 14 months. It has to happen. So essentially, there's a push, essentially. And then, of course, my group members, and the core facilities operations team, and yes, Katie, Pat, Marissa, and Nairi, who are all here. Okay, so thank you. and thank you all. All right. Agnes is after me. That's you. Let me, should I just disconnect? Yeah.
Just want to say that it's the first time I'm hearing about the harassment thing. Oh, yeah. Thank you. Should I move his? Thank you. So my name is Agnes Viale, and I'm the director of the core facility operations at MSK, Memorial Sloan Kettering Cancer Center in New York. The story I'm going to tell you today happened when I was the director of the genomics core. And it's the story of an assay that we developed, that is called MSK Impact. I have no disclosures. And I'm going to start, in the interest of time, I'm going to start my story back in 2002. There was a seminal paper that was published by Mike Stratton and his group that showed that for the first time, we could sequence a lot of genes in a lot of cancers. And that really got a lot of people thinking at MSK. Of course, we're a cancer center, and this is something that we should be doing. Could we sequence all of our patients, et cetera? So in 2006, there was a core that was created, and that was the MSK Translational Oncology Core. And their function was to do amplicon sequencing on the Sanger platform. This is also the time where next-generation sequencing technology exploded. I mean, does it ring? Oh, okay. It will ring here, right? Okay. But not you guys. Okay. Yeah, so next-generation sequencing, exploded in 2006. So jumping up to 2011, my colleague, Nicholas Sochi, who was the director of the bioinformatics core, and myself initiated the MSK whole genome sequencing group. And the idea was to select a small number of samples that would get sequenced for whole genome sequencing, and they would, institutional funds would be provided for the sequencing of those samples. And the conditions were that we, of course, wanted to sequence some MSK patients, but they had to be intriguing cases, so unexplainable response to treatment or resistance to treatment, et cetera. And the case that was chosen was this 70-year-old woman with metastatic bladder cancer, who had a complete response to everolimus, who is an mTOR inhibitor, and she was on a clinical trial at MSK. And only 2 out of the 45 patients enrolled had a complete response. And more than 10 years later, still had no evidences of disease. So this case was sent for whole genome sequencing, and the sequencing revealed a mutation in the mTOR pathway that convey response to everolimus, and this was published in Science in 2012. And the last author of this paper was David Solid, whose name you're going to hear a bit more during this talk. So this was really the proof that we needed to include the genetic makeup of patients in the enrollment of clinical trial, because seriously, 2 out of 45, that's not good enough, right? So had we known her genetic status before, the enrollment could have been improved. So this was for us really our entry into precision oncology. And I just want to highlight that 4 members of core facilities were co-author on that paper. So we jumped to 2013, and something else happened at MSK. Jose Baselga was hired as the physician-in-chief, and he had a very clear vision, which was to bring clinical sequencing at MSK. And he was on a mission to do it. And we were lucky enough that Jose was able to raise a major gift of $100 million from Marie-José and Henry Kravis. And in 2014, the Center for Molecular Oncology was created. So David Solid, who was the last author of the paper, became the inaugural director of the CMO. Mike Berger, a computational biologist, and myself were named associate director of the CMO. And this also was the time where the genomics core, which I headed, and the translational oncology core merged to become the integrated genomics operation that we call IGO, and Niman Mohibullah, who is here, is now the leader of IGO. So, sometimes the stars are aligned, and this is, sorry. I won't tell you why I'm laughing, because it's a little bit embarrassing. But Mike Berger was a computational biologist, and he also had an expertise in targeted sequencing. IGO had the capability to automate target enrichment, and we had clearly financial support, and we also had a strong institutional commitment to make this a reality. So the next 3 years, between 2014 and 2017, we started an intense collaboration between Mike Berger's lab, the integrated genomics operation, and the molecular diagnostic lab. And this is when a lot of creativity had to happen. The fruit of our labor was an assay that we call MSK Impact. IMPACT stands for Integrated Mutational Profiling of Actionable Cancer Gene, a very smart acronym that required a lot of creativity, clearly. And what is IMPACT? After the patients are consented, we collect tumor and blood. DNA is extracted from both samples. We make sequencing libraries, and the exon for 505 genes that are actionable, so there's actually drugs to act on those genes, are sequenced on through next generation sequencing at a high depth, 500 to 1,000x. There was also, during those 3 years, a very intensive work to implement a bioinformatics pipeline. And after analysis, the cases are reviewed by molecular pathologists and released to the clinicians, signed out to the clinicians. So, as I said, during those 3 years, there was a lot of back and forth, and we had really to think outside of the box. And so I made this creativity pipeline here. And if you think about it, it starts with the concept. So Mike Berger in his lab, were big believers or targeted sequencing before it was actually a very common thing. But they could only do small-scale experiment in their lab, right? So in his lab. So if you think that we're going to sequence all the patients, we need to scale up, we need high throughput, we need automation. And that's where the core came into function. So we work with Mike to implement, or to build the infrastructure that we needed, but also to transfer a small-scale assay into a high throughput. And then once we had all the conditions figured out, the molecular diagnostic lab mimicked the platform that we had in terms of automation, software, et cetera, workflow, and they secure regulations to get New York State approval and also FDA approval. And this was really a lot of interactions and a lot of exchange of ideas between those three entities. Okay, so we have a clinical assay, that's great. What's next? And the thinking was really to maximize the scientific return on our investment, right? So we are very lucky at MSK, we have generous philanthropists, but there's no reason that we should keep all the benefit of that for us. So it was really something that was dear to Jose's heart and also Dave Solid. So we really focused on creating a culture of team science. And like I said, Dave Solid, oop, goodness, sorry. Oh, where am I? Oh, okay, hold on. I'm gonna go through the, where am I? Stars are aligned, here. Clearly they were not aligned today. Oh, okay. So David has been, from day one, a relentless advocate of data sharing. He would say, you have the data, we share it with everybody at MSK. And to share the data, you need a data sharing tool. And so one of the tools that was developed is the CBio portal for cancer genomics. And a lot of the money that was given by the Kravitzes went into supporting the CBio portal. And the CBio portal is a repository of all the MSK, the clinical sequencing cohort. So, you know, it's over 100,000 cases right now. And you know, it relates, it makes available to all our clinicians or all our scientists the results of the clinical sequencing. And you can choose whatever cancer type you're interested in. And there's, I mean, I'm only showing you one frame, but there's so many layers of information provided. The return on that investment, and I think this is also part of the creativity or the vision to see what could be the benefits of a culture of team science. We've had more than 700 publications by MSK investigators, thousands of patients that were enrolled on clinical trial now, knowing that they have a better chance to respond to a given drug, given their mechanisms. New drugs were approved, and this was really a key enabler of some of our major strategic initiatives. The second tool that we needed pushed us a little bit further in the sense that, okay, we send your sample for sequencing, and you know, what does, our clinicians are embedded in this talk of clinical sequencing, et cetera. How about the clinicians in community? Do they have the same level of information? How do they make decisions, right? So this was something that we really wanted to address also, and for that we created a second tool which is called OncoKB. So if you think about precision oncology drugs, in 1998 there were 3 of them. In 2023 there are more than 90. And the sequencing reports are you know, really complicated to analyze. They have mutation, translocation, amplification, deletion, copy number, MSI status, tumor burden. There's a lot of information, and unless somebody is trained and used to review those reports, it can be a little bit overwhelming. So they also have to find which drivers are which driver mutations are driving the growth of the cancer, and which genetic alterations are actually clinically actionable, right? And so the biomarkers are generally categorized in 4 categories: standard of care, investigational, hypothetical, or conferring resistance. And within all those categories, there are some drugs that are FDA approved, some, or biomarkers that are for sure going to provide resistance to therapy, some of them for which we have investigational evidences, and some for which we have only hypothetical evidences. So what OncoKB does, it's really a precision oncology knowledge database. It's a repository of all the human genetic variants, and it categorizes the variants and associate them with a potential drug, in response to the genetic modification. This is a fully, this is an effort that is entirely funded by academic, it's an academic effort. And all the data in OncoKB is fully curated by humans. Before OncoKB existed, people worked for 5 or 6 years to curate all the data that was available in the public literature. They work with company. This was a major effort, and it's also the only oncology database that is recognized by the FDA. So the next step was, what more can we do, right? So we're helping our MSK patients. How about other people? So we created several programs that are supported by philanthropy. Again, trying to make the most out of the Kravis's gift. And we have this impacted program to address health disparity, where we have partnership, and patients from Queens County and Kings County in Queens and Brooklyn are getting MSK Impact for free, including the access to clinicians. We have also a program that is called Make an Impact, that offers free MSK Impact to patients that have been diagnosed with one of the 6 very rare cancer anywhere in the world. And so, again, trying to push the boundaries of who else can we help. And this is a picture of a young woman in Africa that benefited considerably from this program. And you can see a very dramatic response in 3 weeks under one of our clinical trial agent, LOXO 101. So it's been now 10 years. 10 years that the CMO was created. Actually, we're getting into 11 years. And so we, of course, looked at the financial return on investment. The first 3 years, the MSK Impact was fully supported by philanthropic funds. And then insurance companies started to bill or recognize or accept billing for clinical genetic testing, and now it's fully sustainable. We get reimbursed by company, but that's not the only benefit from MSK Impact. We have benefited from extramural grants, of course, clinical trial contract, sponsor research agreement, licensing and technology transfer. This is new, a partnership with Sophia Genetics that is going to be offering MSK Impact to whoever wants. And in terms of our MSK's reputation and recognition, we have those tools that were FDA approved, and about 60% of the data that is in the CBio portal is now available through the AACR Genie effort. So the 60% of the data is now available to anybody else in the world. And you know, this is I think a really good way for us to give back to the community, because we've been lucky with all those funds. And this has been also extremely helpful to recruit faculty, of course, writing grant, partnership, and to get more philanthropy, because who doesn't want to be the next Mrs. and Mr. Kravis, right? And I'm just very happy for David and Mike, because they will be receiving at AACR a Team Science Award, and this could not be more deserved than those two folks who have been really focused on giving back and working all together. So this is my last slide, and I'm going to describe briefly what I think is an ecosystem of creativity. And I'm starting with the cores, because I like to put the core facilities at the center of everything. You know, this is where it started, right? Nothing of that would have been possible. It would have been much, much more difficult to do if we didn't have IGO, if we didn't have the genomics core and the translational oncology core. Of course, there's a lot of partnership that had to be built. We know we needed administrators, computational biologists, of course, the business people, the FDA, and the patients. But let's not forget that we are a part of this ecosystem, and we should really take advantage of that as much as we can, or contribute to this. And this is my last for real slide. There's a lot of people to acknowledge. Of course, our philanthropists, the MSK leadership, my colleagues from many different cores, the clinicians, the computational biology, the IT people, and of course, as always, the patients and their family. Thank you. So should I plug in? Yeah.
That's work. It's, we need one of those. Okay. Thank you. Yeah,
Here you go, sweetheart.
I can't see a thing. Right. And
So thank you all for showing up. I'm going to try and make this brief, because we would like to actually have a discussion with you guys. In this environment, creativity, especially at an institution unlike our own that we have very nice philanthropy at, is challenging, and we anticipate it to get more challenging. So I would like to have that discussion with you guys. But let me frame this a little bit. So what's the role of a core director? And actually, after I sent in these slides, I changed it because I saw Shana Mache and remembered that she's the first one I heard call us core scientists. And I think if we look at ourselves other than core directors, core team members, core managers, technologists, specialists, right? We are scientists. And if we reframe that in our brain, it might help us to think a little bit outside the box. So, talking about the box, we do need to know what our box is. There are rules and regulations that we need to follow. There are job descriptions that we have. There are deliverables that we need to make. We also need to keep our teams inside that box. Protocol drift is a problem. not following rules and regulations is a problem. But I do believe that as core scientists, we should be shepherding each other, right? To think outside the box and not to go into that factory mode. That is one thing that I find very challenging. Don't think I've ever done it as successfully as I'd like to, but at least in my team, people tend to get into that factory mode and not think outside. And thinking outside is good. Don't just run, but think outside, and then we'll do it together. And who? So I just spoke about my own team members. I've been running my core for over 25 years, so there's a few of them over the years. The researchers in their projects. So I run an antibody discovery group. That's our cornerstone of the core facilities. So these are development projects. You want to create something new. So that kind of lends itself to the necessity for creativity. Creativity, whether it be in how to get someplace or how to get people to someplace. We start off our projects with, in an ideal world, how should we do it? And then we tamper that with reality. What can we make the protein? Can we do this? Do we have this equipment is certain equipment accessible to us? Do we have the proper controls, right? And then we tamper that down to reality. Other cores, and that's what I'm going to focus on in the next couple of slides. As well as institutional groups. So the Office of Technology Development, we're very fortunate to have them, but sometimes we don't always speak the same language or have the same goals. How to help them hear us in the support that we need from them, and how to hear them and understand what their needs, and come up with creative solutions between the two of them. Where are we? Okay, and so why do this? For me, there's two reasons. And other people in the room may have others, but to me, I want to maximize my tax dollars. My tax dollars fund the research at Sloan Kettering and my core, and I want to get the biggest bang for my buck. The other thing is professional satisfaction. I'm sure a lot of you would love to be involved in more creative endeavors, even if it's not as big as something as an impact analysis. But it does fulfill me to do something other than just churning the wheels. Core creativity, cross-core creativity. So this was a big example, and we were fortunate enough to be funded. I'm pretty sure Isabel got it from our Office of Technology Development. They have a special fund for this type of stuff. So it was initiated by Isabel, who's the former director of our cellular therapy core. And what she wanted to do is she wanted to translate, not from the bench to the bedside, but from the bedside to the veterinary community. And in order to do this, she worked with myself and Ouathek to leverage the fact that rituximab is a great treatment for, it's a monoclonal antibody, and it's a great treatment in humans for rheumatoid arthritis and several types of cancer. I guess other autoimmune diseases that I didn't list. And so basically, this is the structure of rituximab that's on the cell surface of your B cells. And what we did, we knew where the epitope was, we knew where the antibody bound, but we needed Ouathek to figure out how to create that immunogen that we were going to inject into mice. And then we created monoclonal antibodies in my core that were then validated in Isabel's group. And out of this, we got a patent. I haven't gotten any royalty checks yet. It is licensed. Good. Then how come I don't have a royalty check? Oh, $0, okay. But this was fulfilling. This was fun, right? I think it's wonderful that we're able to translate from humans to dogs, actually, because that's what this is against. I apologize. But that's thinking outside the box, I think. Another one is I have what I hope to be a solution, and I need a launching pad. So this one's in its infancy, right? And I've been talking about this and talking about this, And we'll get to the reason why I haven't gotten it done yet in a minute. But either one of you in the room is going to tell me there's a solution, and then I don't have to worry about being creative in this area. Or maybe one of you, and I'm pitching to you, Ralph, can help me make this happen. So we have a problem. Besides developing brand new monoclonal antibodies, we distribute cell lines that were developed and or modified at Sloan Kettering to the outside world. Promotes rigor and, well, promotes reproducibility because we make sure that the cell lines are good, and some graduate student or postdoc are not just sticking them in a box and sending them out. One way of authenticating the cells is through traditional STR analysis, which is wonderful, but it is far from complete, and it does not address all of our problems. Oh, I did go ahead, sorry. So, if we have tumor cells from the same patient, one from the primary tumor, one from the metastatic tumor, they have the same STR profile. Or, if I have a cell line, how many HEC293 cell lines are out there that are derivatives, right? How many cell lines have you guys generated and made derivatives of them? They all have the same STR profile. I can't tell the difference between them. So when I'm sending these cells out, we go through extra steps to make sure that we're sending the right one, and then we cross our fingers because we have no way of confirming this to the degree of precision that I would like. So we have a laboratory at Sloan Kettering that has developed a lot of cell lines where you take the cells. These actually are from MD Anderson, made back in the 70s. They modified them to include luciferase so that you can do in vivo imaging. They inject them either intracardiacally or intravenously, and they find the subpopulation that homes and colonizes an organ. So in this particular example, these go to the bone. We have ones that go to the lung, ones that go to the brain, ones that go to the adrenal glands. So that's 4. I cannot tell them apart from the parent. So when I send them out, how am I really, really sure that they're the right thing? Where did I go? Oh, okay. So let's just go back. The easy one to think about is the primary and the metastatic tumor. They both have the same STR profile. How do I tell them apart? What I'd like to do is to use CRISPR and integrate a sequence, a barcode, I guess, of sorts. And we do this in very sophisticated ways for other things, but this should be possible here where we insert it into the parental one, right? And then you modify it in the same thing and maybe just adding sequence, such as A, B, C, D, F. And I know those aren't nucleotides, but, and then A, B, C, D, E, F met. And each iteration gets a little extension on it. It's a simple way. You can PCR that up and out of cells and sequence it, and I will know for sure what cell line I'm giving out. But I have to find a creative way to do this. And I've pitched it to a few people, and they all sound interested, but why hasn't it happened? Time and money. So this one I don't think will take a lot of money for investment, but I am going to try and pitch it to the institution to see if we, Agnes, I'm going to pitch it to you, who can pitch it to the institution, to see if we can get a little money, because this will promote the reproducibility of the experiments that are being done at Memorial Sloan Kettering. So that's where creativity comes in too. How do you creatively sell this to somebody? It has to meet their needs. Sloan Kettering wants their research to be reproducible. So if we can figure out how to do this, and this can't be hard, it really can't, then we can start doing it for cell lines at Sloan Kettering, right? So that's money. For me, this has been rolling around in my brain for about 1.5 years, has been time. And that's for personal reasons, right? We all have lives outside of this room. But the time, right now, this is not the top priority in my life, but soon I hope it will be. And I encourage each and every one of you to try and make time for creative endeavors, because they do pay off. And like Ouathek said, not everything is going to fly. Sometimes you learn a lot of stuff from your failures, but it's worth it. You're going to make a difference in the scientific community. And hopefully you'll get a little personal satisfaction out of it. Oh, and I got a pretty picture for that, but I forgot to bring it up. Okay. I think we'd like to have a discussion. We don't have the answers. We've sat in your chair, right? We live in your shoes. Maybe you guys have solutions you want to share. Thank you.
So how much money do you need? It's like a fund me something?
Yeah, so for these particular ones, they evolve over time in vitro, so we'll have to find a lot of things to base this on. But thank you for your input, I really appreciate it.
Anybody, wants a recipe? Wants Thank you.
Hello. Hi. So I'm brand new, and I think sometimes when you come into a brand new job to you, you just look at things very differently. So I thought I would just bring this up now to see what people thought. I am planning to get undergraduates from computer science to help with the LIMS system, from the marketing school to help with the marketing program to launch the new core, et cetera. Has anyone done anything like that where and even some graduate students, there are some certain summer internship programs that I learned about that I can do. So I'm wondering if people have done that where you bring in people who have the opportunity to learn things and they can really contribute in that way. And I'm sure there are many pitfalls, but I'm just wondering if we could focus on the positive to start, right? Because I'm doing it. And I'd be happy to report back how it goes. But to me, that seems being creative, taking advantage of the talent that you already have on your campus.
Yep.
Yes, do it.
Do it?
Yeah, absolutely. We don't have a graduate campus. Our graduate school is 6 students or 10, so it's very limited. But tapping into that potential is yeah, absolutely. They, that's all they do. They create. They think.
Yeah.
It's a very good. I also like the second group you want to tap into, the marketing. One thing that you have to think about, in my opinion, is continuity, right? So those folks are going to be here for a small amount of time, and you want a product that is going to survive the departure of any students that you get. So you're going to have to think or put in place a very, support for that to happen.
Which way? There we go. So I would encourage you to. People at ABRF have talked about that in the past, and we're doing it. I never followed up with them to find out how productive an endeavor this is, but I personally think that this accomplishes 2 goals. It gives you people who are interested in performing well to get some stuff done that you might not be able to do otherwise. You can bring in expertise that you don't have. I want the marketing people once you're done with them. But it feeds back to the community, right? It gives them an opportunity to get mentored and to have a good letter of recommendation. So I applaud you for your efforts.
Thank you.
Yeah.
May I say one more thing then?
Yes.
So I'm also, there's a sustainability fund that is funded by the undergraduates on this campus. And so what happened was my lab isn't ready. So I just have a lot of time to think, right? I'm not doing any experiments. So, so essentially, I applied for this grant, and they really like it, and I think they're going to fund it. And the other reason I really liked it, so it's basically partnering with this company called My Green Lab. I had never heard of it. It's a nonprofit that will certify you, it's basic stuff, it's behavioral change, to really bring down carbon footprint, eliminate waste, et cetera. And I also thought the exact same thing, where I'm going to be paying students. I found, instead of having to recreate the wheel, which I had it was almost like, what is that called when you have evolution and you come to the same thing? There's a program at Penn State that is very similar to what I had proposed, so then I actually took a component of theirs where you pay an undergraduate to do this for different course. Anyway, my point being, it was the same thing you said. I'm hoping I'll find people who are go-getters, and that then I could employ in the future for other types of projects. Yeah.
I can tell you, you've been very creative.
Thank you.
Well, that's it.
Yeah, and I think you could also create a pipeline of candidates towards the core, which is also something that we always, it's difficult. And I would say also you can also create a diverse pipeline of candidates, which would be an extra plus.
So let's see if we can find creative solutions to your barriers to creativity. What are your biggest obstacles to having fun, right? And creating interesting solutions, whether they be, it doesn't really matter where they are. I assume many of you have an interest in creativity since you actually chose this session over others. And maybe we're coming for solutions. And as I was sitting here thinking about all of our talks, we're here cheerleaders, but we're not providing you solutions. Here we go. She's got a lot of time on her hands.
I do have one more. We just, they just opened LinkedIn Learning to us at our institute, so we get it for free. There are LinkedIn Learning classes, which if you can get your institute to pay for them, there are whole things about how to get your creative juices flowing, blah, blah, blah. There are all these classes also about maximizing these kinds of things. I haven't taken any yet, but the ones I've taken so far in LinkedIn Learning have been fantastic. So that's something you could do.
Yeah, that's impressive. Yeah.
Yeah, and that's where I was going to go, that a lot of the barriers that people have are, one, you can't play with your instruments. Someone has to pay for that time. And you can't just use random reagents. Someone has to pay for those reagents. So coming back to money, and especially in probably the not too distant future, money will become more and more of an issue. How do you get your institution to put in just a little bit of money so that you can do protocol development? We did, at MD Anderson, we had a process where there was a grant mechanism, and you applied $30,000. Your core director would pair with a PI. PI would add samples, or sometimes technician time. And then the core director would have the instrument and then the reagents. But you still have to get that money.
You need an advocate. You need a champion. You need somebody who's going to fight for the cause. The department have chairs. We have nothing. These poor folks, they all just have me. So, it's like you have to be, whoever is representing the core at your institution have to be annoying, nagging, but at the same time gain the respect of the leadership so that they're heard. And so, if whatever data you can provide, so for example, in our case, the impact or the Center for Molecular Oncology is used as a tool to raise more money. Because I was not joking. The donors, they want to be the next Center for Molecular whatever, right? So you just need you need somebody that is really fighting for the course, and I think it's going to be more and more the situation. Yeah.
Go ahead, Ron.
The gentleman who's going to ask a question, he was my former roommate.
And then she gets the mic.
In my postdoc days.
Okay.
Okay, so I was told that I had to bring a little levity to this one. So time and money is the problem. So all we need to do, since we're all technology people, is learn how to clone ourselves. It's really simple. Okay, got the time problem solved. Money. I saw someone who was a former roommate of mine who had $2 billion a year in sales. He'd gladly,
Now I know where to
will
go.
gladly support. He'll be a philanthropic person and just support all of your creative efforts.
Okay, I actually do have something that might be, so I'm just started thinking about this, like where, what I'm about to talk about was not really a core, but it is, we ran a COVID lab for 4 years. And as we priced things, we basically priced in some protocol development because, the way that we were doing wastewater surveillance, clinical sequencing, the protocols kept changing all the time and we had to keep up with them. And so we just put that in the contract that we had a certain markup on our per sample cost that was really for that. And so, as we now transition into being more of a core, that would be one thing that I would look at is like, a lot of what the faculty want us to do in a core is to help them figure out protocols. So maybe there is actually a protocol development budget that just goes along with being a core if that's really important to you.
So one thing I do in our core is I'm not looking to make a big splash. I don't need $2.4 billion like Ouathek. I want to make incremental steps. Right? And so when I have a researcher who has a project who would benefit from an idea like an immunization strategy or something or a screening strategy, I tell them, I said, as Ouathek well pointed out, one nanny, 2 children, or 1 nanny and 1 child is about the same cost. No, if I was the nanny, I'd ask for more money if there were 2 kids. But anyway.
25%.
25%. So, but what I do is I tell them, if you do this, then I will do this part for you for free. Because 1, I get a controlled experiment. Did this work well? Did this work better? Did this work worse? And then if they want those immunized animals, then they pay for the stuff moving forward. They can only be small little things, right? But I add it in. I kind of slowly build the core up, bit by bit.
You can go ahead, Maris.
I was just going to ask how you, in my opinion or in my experience, creativity requires a lot of time, just for yourself. So being managers and leaders of other people I imagine it can be very difficult trying to find that time for yourself. So how do you sort of block time so that way you can think about things to do things creatively?
I have my answer, but...
Go ahead.
Personal time, rest, vacations, overrated.
That goes with the harassment.
He has no work-life balance. I'm warning you guys.
Exactly.
Yeah, I'm not very good at that neither. I think I feel like there's a lot of there's always a crisis to respond in one of the 26 cores that we have. And so I used to block on my calendar, time, block of time where, I see Nairi just, it just goes away because there's always something happening. So I don't know, Marissa, I don't have the answer. Can I just go back to the previous question? Absolutely. Because I think what I've seen in the course that are managing to devote some of their time and efforts to R&D or, I think creativity, R&D are a little bit the same, right? You need a certain volume of service that, is going to bring you money and subsidize the R&D. So the cores that have enough work to do that, they overprice a little bit the services, and then use that money to fund their R&D efforts. That seemed to be working well.
Hi, I'm James from University of Arizona. Just had a little tip, and this is something that we've done. Try to find clinical laboratories, people who are not necessarily going to go through all of their kits before they expire. Obviously, if they're running a lean operation, that's not necessarily going to be something you're able to pick up. But if you would like to have, basic benchtop supplies and other materials that you might be able to hand over to students for a, sort of a blue sky, let's just see if this works type project. They've already been validated, proven to work. They're excellent quality. They're obviously expired. This wouldn't apply in a clinical research situation, but for us on the just experimental research side of things, it's a good source. And if you happen to have people who have matriculated from your lab and are in new places, keep those contacts up. Hey, what do you guys have any Illumina stuff you're not using? So.
Yes, that's a very good suggestion. Thank you.
Thank you. Ron, you want to?
You can hear me?
Yes.
Yeah.
Okay, perfect. Nice speaking of classroom about the size. So on a more serious note, being the editor-in-chief of JVT, which is the official journal of Data Threat, I would like to point out that as you do these little studies, and you come up with a better way to do something, you should publish that so that everybody else in this room doesn't have to publish it. And while you're doing that, to save yourself some money, don't pay that $5,000 to publish it somewhere else. Publish it in JVT. There are no page charges, and we're open to publishing. So publish for free. Everybody can read it for free. It's published by that central.
Good.
So just one last thing, because Ouathek had mentioned that getting money from the patents is, I know that Agnes has been trying to work with OTD to see if we can claw back, feels like what it is, but we would never phrase it that way to our Office of Technology Development. But when a core contributes so much to an invention that gets licensed and ideally actually makes it to the bedside, which is our mission, that the core should be, subsidized to some way for them to keep doing that moving forward, to make it better, to create a better environment. So that's all philosophical. I'm not sure we've made any.
So I've been discussing that. So there is a plan at Memorial with the sharing. But unfortunately, the bad news is, in my next patent, if I make money, I will get something. But what's done is done. So there's no, I think, core share.
Yeah.
But we still have to work on that, and we still have to push it. You understand? Because to me, I'm not interested in my core getting the money.
Right.
I'm more interested, actually, all cores. For
Right,
example,
all
at
cores,
Memorial should get a share.
Yeah, actually, I'm Matt Kaplan, University of Arizona, Tucson. I have actually noticed that at our university, their visions of patents have changed a lot in the last number of years.
That's wonderful.
25 years ago, when we were involved in some very patentable direct-to-consumer genotyping, they told us that seems silly.
Yeah. You wish you
Has
had.
anyone done the internet DNA thing? Yeah, that we get nothing at the U of A. But anyway, now they do want to patent a lot of things. I've actually patented 2 different things in the last 5 years. And they do say after their patent office gets whatever monies they need, some of it will actually be going to our home department, which in our case is the cores.
Yeah. And the
And
inventors.
someday I might get a nickel.
And the inventors. Yeah.
Yeah, maybe.
Well, that's
No,
wonderful.
I'm not holding my breath
No, a
on that
lot
one,
of
but
people are
if
very
it went to the core,
I
it would be
think the percentages and things are evolving.
And I will say also, like, for all you creatives, it is a new, or maybe just for us, new metric that the U of A wishes to compete on. We're now like number 30 in whatever for patents this past year.
Yes.
So where are you again?
University of Arizona, Tucson.
Okay, great. We have an example, a success story, to end on that note. We can wish, we can hope. Yes, we're at time.
Thank you very much.
Thank you all for being here.
See you soon.
Hi. Very well, and I know she's gone. Can you tell me why? Because you weren't lost, did not tell me all that was happening. I'm gonna walk away. Thank you, Ouathek.
Thank you, Agnes. Not, it was like, it was totally out of the box. You understand? So it was like, Ron, meet Agnes.
I'm sorry, I had to.
Ron, yes. Yes. Yeah, exactly. How about that? Yeah.